Welcome to my world. Another day, another time, another period to expose knowledge. Health is wealth, knowledge is power. And my effort today is to talk about rabies. WHO has given itself 2030 as the year of total complete eradication of rabies. This will be possible if everybody acquired the knowledge of how to prevent it, or even what to do when it is experienced or bitten by a rabid dog or animal. Rabies generally is a virus, and all viruses are difficult to strictly classify as organisms, but because of their genetic makeup, they are able to reproduce themselves using the apparatus of the organism they have infected. So the rabies virus resides in the brain, and that's why it exhibits that madness in dogs and those who are really who probably would experience it, the fear of water and what have you. But we cannot know more without really getting exposed to more knowledge. That is why today's topic is rabies explanation and diagnosis. So the effort I am going to make is trying to expose more what rabies viruses are, and also talk about how to diagnose rabies. If you like what you are listening to, press the like button, subscribe, and you'll be notified any time that there is a quality production out. Rabies explanation and diagnosis. Let's start with the outline. Introduction, WHO key facts and targets. Explanation, diagnosis, prevention. Introduction. Rabies is regarded as the most lethal of all infectious diseases because it affects the brain, and you can understand the brain is just the most, you can say the most important organ in the body. Caused by rabies virus with family name as Rhabdoviridae. The genus is Lissa virus. Another come from the rag or madness. The Lissa means madness, and usually transmitted to man from the bite of mostly rabid dogs. It is bullet-shaped, consisting of single-stranded RNA genome and viral transcriptase, that is the one that really transcribes and let it reproduce its own RNA all over again, surrounded by a helical symmetry called nucleocapsid. Louis Pasteur developed the first man-made viral vaccine in 1884. That was a very important feat, which has efficacy in saving the life of a peasant boy bitten by a rabid dog. You can imagine how important that finding, that discovery was then. Rabies virus multiplies in muscles and connective tissues and passes along sensory nerves to central nervous system, and multiplies and spread through peripheral nerve trunks to the salivary gland. You see the dog salivating when the saliva coming out more profusely when the symptom is becoming very obvious, and at that stage the viruses are in that saliva, and once anybody is bitten, they sure transmitting the virus to that person. This is just to explain the rabies virus. When it enters into the body, it moves, like we said, from the peripheral nerves into the CNS, breaking through the blood barrier and entering up to the brain. At that area it multiplies. How does it really get to multiply itself? it adsorbs to the cell walls and penetrates. These are just the shape, very bullet shaped like. When it penetrates, it uncoats because it has a coating, and the nucleic acid is exposed, and from there it can be transcribed. You now have the translation to produce the covering, the capsomere, and the nucleic acid, and then on to replication and assemblage and the budding of just to get outside of the host cell receptor. The host cell receptor is from where it's adsorbed to the cell itself. And this is just to show that when the dog bites, the movement goes through the skeletal muscle, peripheral nerves to the CNS and to the brain side, where it multiplies, and then it can also affect, move to the salivary gland in multiplying as well. The problem of incubation can be understood when the distance of the bite is far away from the brain. It takes a longer time. Instead of the usual 10 days, it might take up to 30 days or more to show the symptom. But if it bites around the shoulder or very close to the brain side, then obviously the incubation period will be shorter because of the shorter movement to the brain. This is the shape of the virus. It's a very bullet shape, you can see, and this is the electron micrograph showing the bullet shape of the virus. Then what you are seeing inside is the ribonucleoprotein. WHO key facts and the targets of WHO. The rabies, one fact is, rabies is a vaccine-preventable viral disease which occurs in more than 150 countries and territories. So it is widespread, but it has reduced in incidence. The incidence has really reduced. Dogs are the main source of human rabies death, contributing up to 99% of all rabies transmission to humans. Rabies elimination is visible through vaccination of dogs and prevention of dog bites. So if the dogs are vaccinated, if you see any dog that is not vaccinated, then you need to tell the person that please vaccinate your dog, because it can be bitten by any infected, warm-blooded animal, even bats, and then it carries that virus, and it's usually transmitted to man. When it gets the symptom becomes obvious. Infection causes tens of thousands of deaths every year, mostly in Asia and in Africa. That's developing countries. 40% of people bitten by suspect rabies animals are children under 15 years of age. Immediate, thorough wound cleansing with soap and water after contact with a suspect rabies animal is crucial and can save lives. WHO has the target of wiping out rabies by 0 human rabies deaths by 2030. That's the target. Now to diagnosis. Any dead or dying animal suspected of having rabies should be subjected to laboratory diagnostic tests. The technique involves sending the head of the suspected animal to the lab in an ice-filled container or submerged in a container filled with glycerine. The diagnosis of rabies is based on the finding of intracytoplasmic inclusion bodies called negri bodies in nerve cells. You will see is very diagnosis. Demonstration of negri body in brain tissues. The usual practice is once the dog or animal is infected, the virus moves to the brain, and that's where you can easily demonstrate the negri bodies, and that's why it is the brain, the head of the dog, that is usually harvested to do the staining for negri bodies, the inclusion bodies. Take impression smear of that brain and then stain with stellar basic machine, and the results, negri bodies are cytoplasmic inclusion in the nerve cells. So, we move. Isolation of virus by mouse inoculation with brain cells, CSF or saliva, and immunofluorescent test in brain tissue of infected mice. That's one of the other ways of diagnosis. Diagnosis in animal too. Take the specimen, watch death, and brain tissue of the vertebrate animal like earlier said. Collect one PC to Zenga fixative for demonstration of neckline body culture. Egg inoculation, egg embryo, and dog egg could be used. Tissue culture could be the brain, heart, kidney, cell line, cheek embryo fibroblasts, and the diploids, human diploid cell, the primary and continuous cell culture. Molecular tests can also be done. This is real-time PCR. Is now the test of choice for a quick diagnosis of rabies infection. It can be applied to samples. Prevention. Pre-exposure prophylaxis. People who are regularly at high risk of exposure, such as vets, laboratory workers, animal manager, and wildlife officers, should be considered pre-exposure prophylaxis by active immunization with the cell culture vaccine. Where they are even infected, you can contact the health provider. The post-exposure, where it is the animal, in cases of animal bite, dogs and cats in the endemic rabies area should be observed for 10 days. At times they say 14 days. If it lasts up to 14 days, it's considered healthy. If signs develop, they should be killed and their tissue examined in the laboratory. I've told you of the diagnostic test. Vaccines are prepared from nervous tissue of animals. Nervous tissue of animals, fowls, prepared from infected rabbits, spinal cord cell culture vaccine, and many other types.